So we just spent some time talking about how proteins can impact the activity of RNA polymerase, either from a binding standpoint or from an initiation standpoint. Gene accessibility, though, is one of the unique things we see in eukaryotes that we don't see in prokaryotes. We know that DNA is tightly packed using chromatin, which is that DNA protein complexes, which we use histones and establish nucleosomes, etc. So please go back and review the structure of the chromosome if you don't recall. Well, it's this packaging, this chromatin packaging, that ultimately has the ability to impact gene expression. If the DNA is tightly wound and it's packed, and you cannot change it from the open to the close, from the close to the open conformation, you're not going to be able to expose the template for RNA polymerase to read. It's only until you loosen those complexes to a more open conformation in which DNA can now be read. So we spent a lot of time talking about how activators and repressors can block RNA polymerase directly. What we really haven't established much is how these proteins can also impact the level of organization of your chromatin. So here we have multiple enhancers. We can see that this enhancer, based on the DNA modeling, is not available. It's tightly packed, along with the TATA box and the transcription start site. There is no way for RNA polymerase to efficiently transcribe this gene in the present state. But now the actions of a transcriptional activator at an available enhancer site can change the conformation of the chromatin. Now, how does it do that? Well, we know histones are those proteins that are very important in tightly winding the DNA. Well, there are enzymes called histones acetyltransferases, which regulate the activity of histones through the addition of an acetyl group. And so once I modify those histones, the region is, as it says here, remodeled, making it less complex. Now this enhancer is accessible, as well as this core promoter, which allows for transcription. So in this case, it's another example, modifying proteins, histone proteins, by the addition of small groups can change the structure and the function. By adding acetyl groups to the histones, they can't bind DNA with the same affinity, and the ATP-dependent chromatin remodeling enzymes then come and loosen the DNA. So this is an energy-dependent process. And what's interesting is there's actually a code contained within the histones that are extremely important. Many different amino acids in the terminal tails of the histones are targets for this type of acetylation. So a lysine, specific lysine at position 5, a specific lysine here at position 9 are targets of acetylation in the histone protein H2A. There are serines that are targets of phosphorylation. We could see P's here. And so the order of amino acids actually has meaning in those histones. And based on the identity, for example, lysine and arginine have amino groups in their chains. If you go back and look, serine has a hydroxyl group in its R group. So not only the identity of the R group, it's also the position which it's found within the tail has meaning. And it's those amino acids that specifically act as targets that allow the cell to change the structure and the compacting of the chromatin. Let's talk about methylation. There's an enzyme called DNA methylase that adds methyl groups to specific targets. These are common in some eukaryotes. If you look in mammals, for example, 5% of the DNA is methylated. When you add the methyl groups, it causes the DNA to be in a closed conformation, which ultimately will inhibit transcription. We find these cytosine guanine islands near these promoters, specifically in vertebrates and plants that are targets for this methylation. And so when these islands are unmethylated, the genes are on. When you methylate these particular nucleotide bases near the promoter, it turns off gene expression. Now, how does this happen? In two ways. You methylate these islands. It can prevent interaction of proteins like activators near enhancer elements. And so if the activator can't bind, transcription can't be turned on. It also can convert chromatin from an open to a closed conformation. And when it's closed, we know that it's not able to be activated. It's not able to be expressed. And so there are specific proteins that bind a methylated sequence, and they recruit proteins that further condense the chromatin. So if we ask what the effect of DNA methylation is, what would you say? That's right, it inhibits transcription by either decreasing the binding of activators to the region or by keeping the DNA in a closed conformation.